Hi everyone, thanks so much for joining us today. We're going to start promptly at noon Eastern time, so in about 2 minutes. All right, we're going to go ahead and get started. So hello everyone, thank you all so much for joining today. Whoops, every time. Thanks so much for joining today. Welcome to the NOAA Omics Seminar Series. I am your host, Catherine Egan. I'm the acting NOAA OAR Omics Coordinator, as well as the Executive Secretary of the NOAA Omics Working Group, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last several years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside of NOAA. The seminars from this series are all made available on YouTube and posted up on our Omics website. There should be a link to the Omics website in the chat now. As participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation. At the end, I will read the questions for our presenter to answer. And so with that, our presentation today is titled Distribution of Apicomplexans in Shallow and Deep Sea Octocorals of the Caribbean and North Atlantic, presented by Akacia Halliday-Isaac. Akacia, thanks so much for joining us today. I'm going to go ahead and give you permissions to share your screen. So while Akacia is getting set up, I'll go ahead and introduce her. So Akacia Halliday-Isaac is an Afro-Caribbean woman from St. Thomas in the United States Virgin Islands. She's interested in marine ecology and specifically in studying symbiosis and the roles that microorganisms play in various ecosystems. She is currently pursuing a PhD studying ecological and evolution dynamics in the labs of Dr. Colin Jackson and Dr. Peter Z at the University of Mississippi in Oxford. Akacia, thanks so much for presenting today. Just want to let you know that we can see your presentation perfectly, and I'll go ahead and pass it off to you to get started.
Hi, everyone. As Catherine said, my name is Akacia Halliday-Isaac, and today I want to talk to you about an ongoing project that I did with the Smithsonian about apicomplexans in shallow and deep sea octocorals of the Caribbean and North Atlantic. So, first of all, I want to tackle the likely elephant in the room of what is an apicomplexan. So apicomplexans in general are obligate intracellular protozoan parasites. So they have to live within cells of their host. The phylum is thought to be largely parasitic, though there are a few examples of it being mutualistic or commensal with its host. They're unique for what's called apical complex. So at the front of this oblong cell, they have what are called rhopteres and micronemes. Not all of them have all the same pieces, but they in general use this apical area of their cells to invade their the cells of their hosts. So apicomplexans exist in a surprisingly large variety of hosts. So they exist in both aquatic and terrestrial ecosystems in mammals, reptiles, invertebrates, fish, and humans. This phylum contains what I like to call your big baddies. So things like malaria, toxoplasma gondii, cryptosporidium, and most of their life cycles, but not all, involve some type of intermediate vector and a definitive host. So when thinking about apicomplexan life cycles, they generally involved both asexual reproduction, that is called merigony, and sexual reproductions, that's called gamagony. They go through meiosis, create sporocytes, and that tends to be what enters the environment. So when specifically thinking about that general life cycle involving a intermediate host or vector, the merigony portion of it, where they go through repeated cycles of asexual reproduction, is what occurs in your intermediate vector, intermediate host. So apicomplexans within corals specifically have a little bit of a timeline to them. The first ever apicomplexan that was documented within a coral, it was actually something called Nematopsis Nematopsis species. They are more commonly found in bivalves, but between 1984 to 1986, they were first being documented in Porites species, Porites specifically, by Peters and Upton. Later on in 2001, there was apicomplexan protozoan nicknamed genotype N that was found in 2001 in 3 species of the Montastria genus. So it was Montastria annularis, there was a species complex, and then there was Montastria fabulata. There were a couple studies in between then, but then in 2012, one large study, a multi-study analysis came out looking through published genomes from several coral works and finding this genotype N within 16 different scleractinians through just analysis of genome sequences being published. In 2020, Dr. Fossen did a great paper looking at more into the ecology of these apicomplexan, the genotype N apicomplexan. And what he found is it was present in shallow, deep, and mesophotic depths. His paper specifically focused a little more on endosymbiont that these corals have, this coral apicomplexan has. So like a few genuses of apicomplexan, they went through at some point secondary endosymbiosis. So they have plastid within them. And Dr. Vorsten found that that plastid varies by host and depth. So you would find specific plastid types associated with specific hosts and also within specific depths. Recently in 2021, this formerly known as genotype N was officially taxonomically named as Corallicola aquarius by a paper by Dr. Kwong. So overall, this Corallicola aquarius, it's a little bit bigger than Symbiodinaceae within coral cells. It has been found within a vacuole within coral tissues. It has that area up front that has the apical complex. It's found within the mesenterial filaments of the corals, so it's similar to other apicomplexans that tend to stay within the intestinal tissues. Surprisingly, this was found in primarily in seemingly healthy coral hosts. And so far, what we know, so the information we have in the species that has been found in is heavily within the order Scleractinia. A lot of the findings within octocorals, a bit of them came from the Vosen paper and a few studies around there, but they have been found largely in scleractinians, within some black corals and some octocorals. So the information that we have so far, primarily them being in seemingly healthy hosts, has led to the current thinking that they might be commensal with the coral host. But even though they might be commensal, it's important to remember that there are several documented incidents of things that were once mutualistic over evolutionary history becoming parasitic, or parasitic to mutualistic. So it's important to consider them along the spectrum of being parasitic, mutualistic, or commensalistic with their hosts. So specifically, I frame them in the form of like host-parasite interactions broadly. And when thinking about host-parasite interactions, one level to think about them is like within individual hosts, you would look at both the interplays between host traits, environmental context, so the abiotic and biotic factors occurring in their environment, and the pathogen traits. We currently don't know much about the specific life cycle of Corallicola aquarius or its within host growth, whether it's doing any damage to the host cells. On a population level, it's important to think about things like how dense the hosts are, the proportion of the hosts that are infected within a population, the host and parasite, pathogen genetic variation, the environmental and ecological variation within the population, as well as at a metapopulation level, looking at also variation with hosts and pathogens, environmental variations, as well as how those populations are connected. So specifically for my research, what I wanted to look at is specifically the host and pathogen genetic variation, as well as environmental and ecological variation, with an emphasis on octocorals, because most of the work that has currently been done, mostly a lot of scleractinian work. So with the Smithsonian, I wanted to look at the diversity and distribution of Corallicola aquarius within octocorals of the North Atlantic and Caribbean, specifically using museum samples to attempt to answer the questions. One, are these apicomplexans present consistently across octocoral species that are sampled? Are there any genetic variations? Do all the sequences roughly lump together? And does prevalence of this apicomplexan within samples indicate host species fidelity? Does it change with environment, or both? So for my methods, so with these samples, there were samples from the dry collection of the Smithsonian, the wet collection of the Smithsonian, and also some DNA that was lucky enough to be able to use that were already pre-extracted. So with the wet and dry collections, what I would do is go in and get a piece of the coral, a small piece, like 1 millimeter to 2 millimeters at the most, and extract genomic DNA. So I did a couple PCRs, one using a primer that was designed specifically for this apicomplexan, and one that was more general. And those PCR amplifications went through Sanger sequencing. And then later on, what I did is use phylogeny using apicomplexan sequences from various other studies, as well as the sequences that I had to try and get a better look at patterns that may have been going on. So for my preliminary results, I tried to sample a wide array of genera. My sampling ended up heavy in the genre Gorgonidae, Plexaridae, Noidae, and Keratoisidae. And among what I found is I found a few new hosts of the genre Chrysogorgia, Keratoisidae, one new host in Permanoidae, and one new host in Campanellidae. So I was very excited about that, found some new hosts. In terms of looking at the diversity of host species that I found Corallicola aquarius in the shallow depths, so the depths of less than 40 meters, I found only 3 host species, Antilogorgia sp, Swiftia exertia, and Exoracuna. Those had already, the genre had been documented as host genre previously. So most of my excitement came in the deep sea depths, so greater than 200 meters. That's where I found all my new host species, and there were a greater diversity of them. It was very heavy in Plumerella as a host species. but also Genonesis and Keratosis also made up a good bit of the whole species that I found. So my next step, what I wanted to do is create a phylogeny with the sequences that I found. And I also included sequences from others, Corallicola aquarius studies. So 2 Kirk et al. Studies that looked at Corallicola aquarius in scleractinians in Florida. The Boson et al. Study, and earlier Kwang et al. Study that also looked at some scleractinian Corallicola aquarius. So what this allowed me to do is include some more locations and also an additional depth range. So I was able to include samples from the Gulf of Mexico. There were samples from the North Atlantic that were from my Smithsonian sampling. There were samples from the Lesser Antilles, so from the Virgin Islands, from Saba and Tobago. There were also a few samples from Belize, as well as the Florida Keys and part of the Florida slopes. And I was able to look at the samples that came from shallow water depths, so less than 40 meters, mesophotic depths, so from 40 to 200 meters, as well as the deep sea depths greater than 200 meters. So what I found, which was very interesting to me, is I found 2 separate sections of this tree. So the samples that I collected I've highlighted in these blue boxes. But in general, I found one section of my phylogeny that seemed to be not really separated by depth, but more by the host, the order of the host, as well as a second portion of my tree that was much more conforming to depth gradients. So overall, my takeaways from this brief study, fun fact, I did this all was able to do this all in 2 weeks of lab work. It was very much a very fulfilling experience. But I found a greater presence of Corallicola aquarius in my deep sea octocorals that I sampled. And there are potentially within my phylogeny 2 clades of Corallicola aquarius that vary based on the order of their host and the depth that they were found at. So thinking back to overall looking at host parasite interactions, building on this work in the future, I would like to look more at connectivity within these populations and overall within like an individual population, the portion of populations that may be infected. Additionally, looking more at that, I have another study that I'm currently working on that was looking at within a population, not only the proportion of infected, but any microbiome changes. I'm excited to talk about that in the future as well. But this project, like I said, was very recently done. It was funded by the Kenneth J. Boss Fellowship in Invertebrate Zoology at the Smithsonian. Because of that, I would like greatly to thank the Trini lab in general, because I kind of blew in like a storm talking about apicomplexans, and they were amazing, helping me, as well as the LAB at the Smithsonian that also somehow put up with me blowing in like a storm. With that, are there any questions?
Awesome. Thanks so much, Akacia. That was fantastic. It's super interesting. I know for sure we would love to have you back to present on your future microbiome research. So we'll have to stay in touch there. Now is a good time for questions. If anyone has any, you can use the questions box here to ask, and I'll read them out loud for Akacia to answer. So I think we'll just maybe give it a minute, let everybody stew a little bit on the presentation, see if they have some questions that come up. I guess while we're waiting for questions to come in, Akacia, I had a question for you. I hope it doesn't seem too silly because this is not my area of expertise. Could you talk a little more about, you touched on it at the beginning, the idea that maybe the apicomplexans and octocorals are not parasitic in nature, but maybe more of like a commensal relationship? Could you just like elaborate on that a little bit more?
Sure. So, let's go back. So the idea behind Corallicola aquarius potentially being commensal came up partially due to the plastid that it had inside of it. So this plastid had photosynthetic genes in it, which later the theory is just because it went through secondary endosymbiosis, it at one point picked up a photosynthetic red algae throughout its evolutionary history. So that, as we know from other apicomplexans, it could be helping with some metabolic needs, although it's not fully clear exactly what its current role within this apicomplexan is. But in some other apicomplexans, like toxoplasma, their endosymbiont kind of fills in a little bit of metabolic needs that it has. But a major thing that has bearing on it might not be parasitic within the host is that pretty much all the hosts that they've been detected in have been seemingly perfectly healthy coral hosts. So that may be due to it just being commensal. It's obligate intercellular protozoan, so it needs to be within a host, but in rare cases, they don't necessarily have negative effects on the host. But another thing that could be involved in it, which is why I was interested in looking at microbiome changes, is that sometimes microbiomes may they interact with these pathogens to help to mitigate host effects. So it's all a really interesting triangle of the pathogen itself may not actually be a pathogen, or the microbiomes may be helping in some way.
Awesome. Thank you so much. That was really helpful in helping me figure out what this relationship is between the host and the apicomplexan. Awesome. Okay, I have a couple people who are raising their hands. Unfortunately, I'm unable to unmute you. The way the webinar works is the audience members cannot speak. So if you do have a question, just type it directly into the questions box, and both Akacia and myself can see it, and Akacia can answer the question out loud. So I do see folks with those hands raised. Please type your questions into the question box. We'll wait a moment to see. Okay, I had a question, Akacia, that came in from Adela. So they ask, I wonder if you were expecting that C. aquarius was more abundant in host species that live in deep mesophotic rather than in shallow waters.
Hi, Adela. Actually, no, because there was a previous study that their finding was that they weren't finding it in a symbiotic, um, so a zooanthely corals. So I was very surprised by that. Um, so thinking about it, it does make some sense that since they're showing some level of host specificity, um, that the that the depth wouldn't be too much of a deterrent, but it's kind of exactly opposite than what I would have expected based on the previous study.
Thanks, Akacia, and thanks for your question, Adela. I had a question that came in from Esther. So Esther asks, who has done TEM studies of Corallicola species? And can you examine whether the pathogen comes from these species or not? I hope I interpreted that question correctly, Esther.
Um, I'm not familiar with the term TEM studies. Oh no, actually I am. So I'm assuming you mean transmission electron microscopy. So for this specific I probably should stop calling it a pathogen. So the first transmission electron microscopy that I've seen has been the Kwang 2021 paper that taxonomically named the Corallicola. And remind me what was the second part of that question?
Yes, let me navigate to it. And Esther followed up and said, yes, transition electron microscopy. So you got that part. And then they ask, can you examine whether there's a pathogen that I assume shows up in this or not from this technique?
Yes. So it can be used to look for the pathogen. So I believe what Dr. Kwang did is he did both confirming with PCR as well as you can do histology on the coral tissue, which is what he did. I personally have no experience with doing histology on coral tissue, but it is a viable method that would be really interesting to look at in some of the preserved samples. I was actually interested in doing it in the beginning of my dissertation, but then with the pandemic, it did not become a viable possibility anymore.
Thanks, Akacia. All right, and I have one more question that came in from Esther also, and they ask, will you do more field studies, for example, collecting samples from populations of gorgonias in an area to obtain data to look at the host specificity and metapopulation distributions?
Yes, I would like to. I did look, like I said, with the microbiome study, but my current dilemma with that is it's very heavy on one specific species, Gorgonia ventolina. Apparently, Corallicola aquarius really likes Gorgonia ventolina. So I have to gear up to include more species and more sites to get a proper look. I'm biased to the U.S. Virgin Islands because I grew up there. So I'm currently looking into more places to sample, more sites, more species to look at. And also the samples that I took outside of Smithsonian has been all shallow water. So I also would like to look into also looking at the 3 depth ranges that I considered in this study, but at a specific set of areas.
Great, thanks so much, Akacia. I think that's it as far as questions. So I'm gonna go ahead and wrap up the webinar, but if folks have lingering questions, please reach directly out to Akacia and ask. So Akacia, thank you so much again for presenting today. Your work is so interesting. I think it's just a great part of the Omics Seminar Series. We really appreciate it a lot. And thank you to our audience for joining us today as well. The NOAA Omics Seminar Series generally takes place on the 3rd Wednesday of every month. We may schedule some webinars outside this time frame, so just please take a look at our website linked in the chat for up-to-date information on future webinars. Our next one is on February 15th at 12 p.m. Eastern Time and is titled Making eDNA Count Towards Quantitative Metabarcoding, presented by Dr. Zachary Gold from NOAA's PMEL laboratory, and specifically from their omics program. So registration's up on the website, and I'll see you all at the next one. Thank you all again, and thank you again, Akacia.
Thank you for having me.